Welcome, everyone. Thanks so much for making the time to be with us today. We will get started in just a moment. Well, hi, everyone. I'm Michael Morella, Managing Editor of Events at U.S. News and World Report. Today's webinar is Accelerating Bench-to-Bedside Advances in Pediatrics. We're going to be exploring how hospitals are enhancing the relationship between researchers and clinicians to speed the pace of new discoveries, benefiting young patients coping with chronic and rare diseases. For more information about today's speakers and past and future events, please visit usnews.com/events. Today's webinar is also being recorded, and it will be available to watch on demand on our site. Now, to get us started, we're pleased to present a recent conversation that U.S. News Managing Editor Liz Whitehead hosted with Dr. Huda Zogby, Founding Director of the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital. Let's take a look.
Welcome, Dr. Zogby.
Thank you. Very nice to meet you.
Around the world, an astounding 1 billion people, including 300 million children, are suffering from the ravages of devastating brain diseases. In 2010, you founded the Jan and Dan Duncan Neurological Research Institute, which you continue to lead. Can you start us off by describing its mission and focus?
The mission of the Institute is to identify as many causes of neurological diseases as possible, understand the mechanism of the disease, and come up with meaningful treatment. That's really what we set out to do and continue to do.
So much of what you're trying to accomplish at the Institute has been informed by your personal experience as a physician scientist, researcher, collaborator, and leader. So I'd like to take you through some of the turning points of your journey now. First, a little background for our audience. In the mid-70s, you came to the U.S. to complete medical school when the civil war in your native Lebanon made it too dangerous to stay. Though other schools rejected you as an international transfer student, Nashville's Meharry Medical College did not. After a challenging transition year, you excelled at Meharry, only to hit another roadblock. For whatever reason, various residency programs declined to interview you. But Dr. Ralph Feigen, then physician-in-chief of Texas Children's, saw your application and invited you in. What happened?
Magic happened, I must admit. After feeling such a challenge to really transition, leaving home during the war and coming to a new country and starting in a new place, I was so grateful that the dean at Meharry Medical College gave me a chance, and I got a great education there. And I was so happy when Ralph told me that I'm welcome at Baylor College of Medicine. And I loved my pediatric training here. I thought I'll be a pediatric cardiologist, but when I rotated on neurology, I met Marvin Fishman and I really became fascinated by child neurology and got my training in child neurology. And the intention was to be a clinician, but patient encounters changed my course, and that's why I pursued science.
Yeah, and the story of your life has been unexpected twists and turns. And you had completed your pediatric residency and your pediatric neurology residency at Baylor College of Medicine. And as you said, by 1985, you're on track to be a clinical pediatric neurologist. But then you met a 5-year-old girl named Ashley who changed the trajectory of your career. Can you talk about Ashley and the path she set you on?
Sure. So I met Ashley actually in my first year of child neurology training, and I was heartbroken about her illness and the course of her disease. Ashley started life like every girl, developing normally, did all the things that 2-year-olds will do, including sometimes mouthing nursery rhymes and using her hands effectively. But all of that changed after her second birthday, and everything she had learned, she lost. And language stopped, social interaction stopped, and instead of using her hands, she started wringing her hands. Her balance deteriorated, and within the span of a few years, practically every function in the brain wasn't quite normal, wasn't quite typical. And I thought that's really devastating, and I was intrigued by that. And then a chance encounter a week later with another girl with Rett syndrome made me really pursue this disease. At the time, the disease was only described in European cases in one report by Hagberg and colleagues. So having seen two in 1 week, I was convinced there is more. And the clinic volunteers, the Bluebird Circle clinic volunteers, helped me pull chart, and I reviewed them and found a few more cases. And that really set the course of my career. As I studied these cases and published my first paper with my colleagues and mentors here, I decided as soon as I finished child neurology, I will go to the lab, learn molecular techniques so that I can find the Rett syndrome gene.
And is it fair to say that Rett syndrome then was understood as a constellation of neurological symptoms, but the cause of it was very much unknown?
That's correct. It was a syndrome that was described in Europe, as I mentioned, in about 35 girls, and before that by Andreas Rett in 1966 in the German language. So it was very poorly understood. Not many people were aware of it clinically. And when the Hagberg report came out and we learned about Rett syndrome and started seeing patients with Rett syndrome, we realized that this exists much more frequently than we thought, but nobody knows the cause. And to me, it was intriguing because the girls are born healthy and then they regress and they lose milestones, but it's not degenerative. And the fact it's not degenerative and not also early developmental, where early on they're doing well, was really intriguing as well as heart-wrenching, as you can imagine. And nothing was known about it, but it was always females. And that to me was a clue that it must be a genetic disorder. Why would it spare the males?
So you wanted to go into research to find the solution. And of course, wanting to pursue research it and accomplishing it are two different things. You approached Dr. Arthur Baudet, a preeminent geneticist at Baylor, to pursue, as you said, postdoctoral research in molecular genetics. The only problem is that you didn't have a research background. How did that conversation with him go?
So I have to admit, Dr. Baudet was welcoming in a sense. He knew me from the clinics. He had encountered me in the clinic and he knew that I'm a good clinician. So when I visited with him and shared with him my interest in finding the Rett syndrome gene and my interest in learning molecular biology from him, we had a very nice conversation. He did give me a quiz in genetics to really make sure I have good fundamental knowledge, and that I passed. And then we talk about the process of becoming adept in molecular biology and molecular cloning and so on. And we discussed Rett syndrome. I told him about my passion about this disease. And I told him I have samples from about 200 different girls. And he was showing me the families. And I showed him that this is a sporadic disease. That means it's only one in a family. And this was 1985, when we didn't really have the genome map. PCR wasn't even discovered. Polymerase chain reaction amplification was not even discovered. And, there was no way. We knew very little about technologies that will speed up genome cloning. So when I shared with him that this is a sporadic disease, he said, I don't think you can work on that. I'm really sorry. This is like a needle in a haystack. Find another problem. And we went back and forth, and he was working on urea cycle defect. He was a giant in that field, but I told him I'm really not interested in that. Interested in different problems. And he was such an awesome mentor who really let me bring neurologic disease research into his lab outside of metabolic disorders. And we identified a family with a dominantly inherited balance disorder. And I told him, this is what I want to work on. And that's what I wrote my K grant on. And he was really supportive. And in hindsight, I'm very grateful for his support, because by working on a C1, I was able to be productive, whereas the Rett research took much, much longer for us to make the discovery.
So can you talk a little bit about spinocerebellar ataxia type 1?
So, spinocerebellar ataxia type 1, or we call it SCA1 for short, is a dominantly inherited balance disorder where the individuals will be healthy till they're about 40. And then that's when they manifest balance disorder, and gradually their balance deteriorates. They'll become wheelchair bound. Eventually, their breathing and swallowing become uncoordinated, and they will choke and sadly die prematurely. And there was a family here in Montgomery, Texas. It's about 50 miles north of town. So I would travel there and examine members of this family and collect blood for DNA samples to map the gene. It's inherited in a dominant manner, which means if a parent is affected, each child has a 50-50 chance of being affected. So I had over 100 affected individuals and constructed a 200-member pedigree. So this is a really Mendelian disease where you could map the gene. And right about the same time I was working on this, Huntington gene was mapped. So we thought this is doable. If Huntington could map their gene, we can map this gene. And with the mapping of the gene, then we pursued to get closer and closer to the gene event to eventually clone it.
So it was still an incredibly challenging problem, but not as challenging as Rett syndrome.
Correct.
So you began to work on these very important rare diseases, and yet within 3 years, you ended up applying for funding to start your own lab. What gave you the impetus to do that?
So I maximized my training in arts lab. I obviously worked at the bench. It was a great environment to learn all various molecular techniques so that you can become adept at mapping and cloning and screening for new markers and so on and so forth. And having the K funding, of course, helped me really focus 100% on the research. My time was protected, so I didn't have to do any clinical medicine for those 3 years. But I felt I needed background in basic science, so I created my own program because I didn't want to be just a geneticist. I also wanted to be a neurobiologist and understand principles of cell biology. So I took quite a few graduate courses in those 3 years. And as I moved on in the lab and worked as hard as I could, Art felt by the end of 3 years that I'm actually ready to move on to my own lab. I wasn't sure I am ready, but he was sure and put that confidence in me and wrote my first R01 and got it. And that's when I moved to my own lab. And it was an exciting time. It was a very small lab, just me and a graduate student and a technician. We were just 3 people moving into that lab, and we grew very gradually and slowly.
I have to think that having your little lab and your little team together, that it made you very close and probably has informed what you think collaborative relationships would be. What was it like with just, was it the 3 of you?
We started just the 3 of us, and I had a lab. I was very eager to be in a strategic basic science building where I can interact with all the new faculty. At the time, Dr. Tom Kasky, the founding chair of our department, was building, it was an institute, now it's a department. He was really keen on hiring people with diverse expertise, mice, yeast, genetics of flies, and variety human, and mixing us all together in that building. And I really wanted to be in that building, but space was tight, and I accepted to take 2 benches in a lab of another recruited colleague and said, I don't need the office. I'm happy just to have the benches and I'll sit at a desk. And I think that was probably the greatest thing, because being within the lab, always working with my student and my technician, who's still with me today, Alana, has been with me all these years, was extremely helpful and productive. I did not go sit in an office. I actually interacted and then recruited a second graduate student and a second technician. And by me working side by side with them, I was the motivator. I was the one with the best hands at the bench. I truly believe those first few years were very critical in my success as a young investigator.
And I think you did something else that was really, really smart and necessary. I mean, it seems to me that you always are reaching for additional knowledge, and you reach out to people even when you don't know them. And you make cold calls to colleagues around the country. And I think you made a very important one when you reached out to Dr. Harry Orr at the University of Minnesota. Can you talk to me about why you called him and
Sure.
what that has led to?
So Harry was this brilliant investigator at the University of Minnesota. He was, I think, associate professor at the time, and I learned and I read papers that he is also trying to map and clone the SCA1 gene. And I thought about it. We're both working on the same disease, and we're both generating critical tools. I was generating tools to help you create new markers for the chromosome so that you can walk closer and closer to the gene. And he had really large families. And I thought if we pool our resources together, perhaps we will get to the gene faster. So I called him about the possibility of collaborating. And I think he was a little bit taken back. He was silent for a moment or two. And then he said, let's do it. He calls this the call. And I think we'll both forever remember that, because it was 1988 when we started our collaboration. And to this day, it's the one thing that we cherish the most, because we both learned so much from each other, from our trainee. We pulled resources. We went into multiple directions, complementing each other. It was really the greatest joy of both of our careers. We both consider that the greatest joy.
So you ended up with a 30-year friendship and collaboration, and it has also paid off. Can you talk about what you have discovered?
So on April 8th, 1993, we got to the point right before, let me back up, before April 8th, we got to the point where we now narrowed the region to about 1 million base pairs. And we know that the gene is in that 1 million base pair. But back then, in the early 90s, 1 million base pair is a lot of DNA to sift through to find the right gene. And Tom Caskey gave a presentation about myotonic dystrophy. Fragile X was discovered a couple of years before that. And then Tom gave a talk about myotonic dystrophy being a trinucleotide repeat, these 3 bases of DNA that repeat themselves. And they pass from generation to generation, and the disease gets worse. And that's exactly what I had seen in my patients. You know, I examined all these SCA1 patients and I saw the disease gets worse. So I called Harry and I said, you know, it could be these trinucleotide repeats. What about we focus just on those and look for them in that 1 million base region? Maybe we'll find one that's expanded. And we decided to split the region in 2, but we kept about 70,000 base pairs of overlap. And on April 8, 1993, we sent each other faxes. We both discovered the mutation on the same day, and it was in that region of overlap. So
just stunning. And it's opened a pathway for treatment.
That's correct. We then spent a lot of time creating different tools to study the disease, to understand the effect of the mutation on the brain, to understand if it's reversible, to understand mechanisms and ways to sort of lower the toxicity of the mutant protein. And that's leading to now new pathways that could be applied eventually for therapies. So that's correct. And we did it together. We kept going back and forth, advancing, building on each other's discoveries and coordinating the research between us. And it was really that first collaboration that gave me such a great taste about collaborations. So after finding the gene, remember, in arts lab, I learned human genetics, I learned cloning, but I've never touched a mouse. I've never done biochemistry. So shortly after you find a gene, you want to understand the gene product, the protein. You want to work now with animal models. And the wonderful collaborative environment around me allowed me to reach to the best expert in mouse genetics, the best expert in biochemistry, and I started reaching and collaborating, and that's how we kept on advancing. This is why I learned to really value collaborations, because you know, the disease doesn't understand the boundaries of our training. The disease affects every aspect, you know, in the tissue. It's affecting the biochemistry, the physiology, the RNA, the proteins, and of course the behaviors. So having those multiple expertise and learning from others and working with others have been extremely valuable. And that was the inspiration to start the Duncan NRI. It is to bring people with diverse expertise and put them together so they can collaborate to solve tough diseases.
What an amazing way to characterize it, that the disease doesn't know boundaries. And so you can't limit the boundaries for what expertise you think you may need to bring in to attack it. So all this time that you were working on these other major research initiatives, you never lost sight of Rett syndrome. And it has turned into a decades-long research commitment for you. Can you describe how you got back to it and where you are now, and how you ended up discovering the gene that had the mutation that caused it?
So really, I never stopped a single day or month working on Rett. Also, although while in arts lab, I was working on SCA1 on the side with my own hands, and then Alana was helping me, and the second person we hired help. We were always trying to find ways to get closer to the Rett syndrome gene. We started by narrowing the genome from the whole genome to the X chromosome, realizing that this affects girls. And the only way I could think of it affecting girls is being on the X, because females have two X's. So the idea would be if the mutation on the X and the girl has mosaic, half of her cells affected, she will have Rett, but the boy will be very severe. So that was the way we've worked on it. And basically, we really tried to walk the X chromosome and test different genes to find the candidate gene. So it was truly a brute force effort after narrowing it down from a handful of families that we identified and collaborated. Also, some families came from Uta Frankas and Carolyn Chen, and we narrowed the region to 30 megabases and simply started sequencing gene by gene. And back in those days, just for the younger generation, when you sequence a gene, you had to first identify all its components, which part make the protein, the exons that make the protein, which parts don't, and you do all the sequencing at the bench. There wasn't any of that, just send it off for sequencing. So any genes that we wanted to exclude took about 6 to 12 months, depending on the size of the gene, at least 6 months. So we're going literally gene by gene, and we had many red herrings. I will tell you, there was one time we found a gene on the X. We were very excited. This is it. The girl had a mutation that inactivated the gene, but then we found the same inactivating mutation in her healthy father. So we knew that it couldn't be. Sometimes we would find a patient with a break on the X chromosome, translocation or an inversion. We spend a year or 2 or 3 cloning those breakpoints, and then we don't find the gene there. So we had a lot of, twists and turns, but finally, it got to the point nobody really in my lab wanted to work on Rett syndrome anymore. So everybody abandoned that project. I had many more excitement over the ataxia project. We discovered a gene that's really critical for development of inner ear hair cells and the cerebellum, and the balance, sensory balance, proprioception. So everybody was working on that gene, atonal and SCA1. Nobody wanted to work on Rett. But Ruthie Amir was a wonderful postdoc who's joined the lab, and for 3 years, really nonstop, she worked gene by gene until in August 1999, she discovered mutations in a handful of patients, and then we confirmed that, and that's how the gene discovery came about.
Which is MECP2?
Correct. It's a gene that encodes a protein called methylcytosine binding protein 2. I call it MECP2 for short. When cytosines are methylated, it can bind and it affects gene expression. That protein was identified almost 9 years before by Adrian Bird as a protein that binds methylated cytosines. But nobody knew anything about its role in brain function. Nobody had any connection to Rett. So when we found it as the gene that's mutated in Rett, now we, started to try to understand the mechanism of disease.
And in fact, I think you're working on two tracks right now. You've discovered that there is the potential for intensive behavioral therapy, perhaps to be effective before symptoms show up with some children, and then you're looking at sort of other treatment avenues. Can you talk about those?
Sure. So one important thing we learned from the various genetic studies and creating different animal models, one thing we actually learned is that one, the dosage of this protein matters. If you have too little, you have Rett syndrome, and if you have too much, you have another disorder. We call it, and then Linda Van Esch in Belgium found humans with duplications of the gene. So we first discovered it in the mouse that too much of the protein can cause disease, and then she found first set of humans, and we saw many here with duplications spanning the MACP2 gene. So we call it the Goldilocks protein. You have to get it just right. And we've worked on that area to try to reverse the disorder and come up with a therapy. I'll get back to that later. But for Rett, we were really trying to understand, why does loss of this protein in half of the cells cause so much havoc and so many neurologic problems? And it turns out this protein is really important for the function of the neurons. And when it's missing or mutated, you partially disable the neuron, where it's functioning at about 70% of its capacity, not 100% capacity. So as we began to contemplate, and it's important for all brain cells, so as we began to contemplate therapies here in the Institute, we had phenomenal neurophysiologists. So I collaborated with a colleague, Jean-Roch Tang, using deep brain stimulation, which, as you know, is used in the clinic to treat Parkinson's disease, some tremors. And we found that if you stimulate the circuit, now you normalize the activity of neurons and you change all the symptoms, at least in the part of the brain we stimulated, the hippocampus, so the animals can learn and can remember. But the Rett affects the whole brain, and we're thinking it's really going to be hard to bring back the normal capacity of function to these neurons if we're going to have to do multiple regions of deep brain stimulation. So we were pondering what can mimic brain stimulation, but it's less invasive. And that's where the intensive training came about. The idea is, as you know, the more you practice something, the more you can get better at it. And we thought maybe the Rett neurons can get better if we do intensive training. But we knew parents do a lot of physical therapy when they have a child diagnosed with Rett syndrome. So we wonder that maybe the timing of that intensive training is important. And if we started before symptoms, it would help. And that's what we did in the animal models. And Nate Akili, the student who did this, found that indeed, if we start early and train them either on a motor task or a learning task, we can get them to behave almost as good as normal. And we will keep it up and delay the onset of the disease in mice by months. So that's really exciting. And if we can sort of identify girls early on, say at birth through newborn screening, we have typically a couple of years, 2 to 3 years. Most girls are diagnosed by 3 years. You have a 2 to 3 year window to train them. And even if it's just delaying the course of the disease by 1 year, let's say, that's great until we have more definitive treatments and they can respond better to any new interventions that we will come up with.
And you, just very briefly, you alluded to a related disease that is affected by the same gene, and the MACP2 duplication syndrome,
Correct.
and which is kind of not the opposite, but it's too much, right? So, and you've been and there are 2 points I want to ask you about. One is, I think that what is so interesting about the research is when you identify a certain gene as causing a problem, you find actually a constellation of other diseases that may be impacted. And so, yes, you start on Rett, but now you have treatment avenues for another syndrome because you discovered this gene and you discovered how it impacts different parts of the brain.
That is very, very true, what you just said. I actually learned so much from studying Rett syndrome. And to your point, let's focus first on the duplication. We've studied that. We had the mouse model that reproduces all features of the disease. So we tried a treatment where we use a small piece of DNA called an antisense oligo that you can basically deliver in the ventricle. You can do it eventually, as you know, in spinal muscular atrophy. Were delivered via spinal tap to human patients. You put it in the spinal fluid and it'll hybridize to the RNA of the MACP2 gene. And if you have too much RNA and you've got this small oligo, now this hybrid will be cleaved by enzymes and degraded. So you can decrease the mutant protein back to normal levels. And you can titrate this just like you would titrate blood pressure medication or blood sugar medication, because you don't want it to go too low. So when we showed in mice that you can actually treat these mice when they're symptomatic, adult mice, we even waited till they're 9 months old where they're having seizures all day long. And when we normalize their MACP2 level, we found you can actually reverse the symptoms and they get much better. So that is really pretty exciting. And that gave the proof of concept data to move to clinical studies, and natural history study is now ongoing in preparation to start such clinical trials building of these studies. So that's really exciting that this kind of work, this in-depth work that led you to discover too much of the protein is bad. You have the animal model, you can reverse it, and now there's at least a strategy for these children who are devastated by having too much of this protein. That's
I'd
one.
like to, go ahead.
Sorry, I just want to add, that's one end of the spectrum. The other end of the spectrum, when Rett syndrome happens, you typically have a mutation that inactivates the protein pretty badly. But we also learned that milder and milder mutation typically cause more psychiatric disorder or just autism, or autism with behavioral problems. So it's sort of, you start learning from one disease and one gene about the spectrum, from the very mild that may manifest later on in life as a psychiatric disease, all the way to the most severe that manifests in the neonatal period with very severe disease.
Well, this is great. I actually, what you just said, I want to actually bring us full circle now back to the Duncan Neurological Research Institute and your vision for it, because as we talked about, translational research seems to depend on both a investment in pure research where you're not sure where it's going to go, targeted research, and also an understanding that one discovery may lead to, for one disease, devastating disease, may lead to solutions for many, but it requires very robust research capabilities. Can you talk about what you tried to bring together at the Duncan NRI, what kinds of expertise and resources to not only propel the research, but also tighten the relationship between bench to bedside or bedside to bench to bedside as your route?
Thank you. Actually, that's a very important question. So if you look at the Duncan NRI, you're going to find among our 33 or so investigators, about 7 of them study fly genetics. They study the fruit flies. And those individuals have in the early years done pure fundamental research to try to understand development, to try to understand how the body is laid out, how limbs develop, how brain cells develop. But through that work, they understood the synapse better. Through that work, they developed new technologies to engineer and manipulate the flies in so many different ways, to the point we can take out any gene, replace it with a human equivalent of that gene, and learn about the relationship between humans and fruit fly function. So fast forward to the past 4 years, that became really the cornerstone for discovering the most complicated, unsolvable disorders. Basically, when you have one patient with a mutation in their DNA that has never been seen before in anyone, how do you know this poses the disease? Here comes the fruit fly to the rescue, where you eliminate the fly gene and the fly will have damage, let's say, to the eye. You put the human gene, you rescue, and you put the human gene with the variant, and if it rescues, you know that this variant is benign. If it doesn't rescue, this mutation is inactivating the gene. That's one example. And about 40 diseases that were unsolvable from around the country came to us, and the team worked together to solve these diseases. So you got a fly geneticist working with a clinician, working with someone who may do human neurons, and working with the biochemist, all of them working together to actually solve a problem and understand it better. So, and then there's, of course, computational biologists that help with the analysis. So it's that kind of environment that we need today to really start moving forward with solving challenging disorders.
And, she said that the human genome wasn't even mapped when you were starting work on RED. And now you can rely on, I think you've got AI and big data and engineering. And just briefly, can you talk about your core labs?
So one thing, we mentioned the need to really approach a disease from so many angles, right? Behavior, physiology, biochemistry, computation. And I know myself, as you alluded at the beginning, I reach out to people. I'm not shy of calling someone cold, but most scientists would not do that. It's a little bit intimidating for some scientists. It's a little bit unusual, but by being surrounded with resources and expertise that you can just walk down the hall and you find someone who can characterize your mouse and do behavioral studies on it, somebody who will do physiology and collaborate with you, it's much easier. So we brought a world to allow intense collaboration all under one roof. And that's what makes a big difference. And important part of that is having computational scientists and engineers. Now we all talk about AI and the power of computation. But my first hire in the Institute was in 2010, computational scientists. And having computational scientists as a mathematician was very important from back then for us. And we grew together. So they speak our language and they understand it, and we go back and forth.
Oh, that's wonderful. Dr. Huda-Zogby, thank you for sharing some of your remarkable story and the inspiring work that you and your colleagues are engaged in unlocking treatments for these neurological diseases and bringing hope to millions of patients and the family members who love them.
Thank you so much. It's my honor. Thank you.
I'm going to invite all our panelists. They're turning on their cameras and audio, and I will introduce them to continue this fascinating conversation. I'd like to start out by welcoming Dr. Sue Firth, Executive Vice President and Chief Scientific Officer of the Research Institute at Children's Hospital of Philadelphia. Dr. Mary Leonard, Professor and Chair, Department of Pediatrics, Director, Stanford Maternal and Child Health Research Institute, and Physician-in-Chief, Stanford Medicine Children's Health. And rejoining us is Dr. Huda-Zogby, Director of the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital, Distinguished Service Professor at Baylor College of Medicine, and investigator with the Howard Hughes Medical Institute. Thanks so much to you all and our audience for joining us for this important discussion. Dr. Zogby discussed her role as director of the Duncan Neurological Research Institute. I'd like to ask Drs. Firth and Leonard now to similarly sketch out their roles at their respective institutions. Sue, can you start off?
Thank you, Liz, and thanks for the opportunity to be part of this prestigious panel. I really appreciate it. So I lead the Research Institute at Children's Hospital of Philadelphia. As you mentioned, I'm the Chief Scientific Officer, and the Research Institute encompasses the research-focused faculty and scientists across the enterprise. So there are over 1,000 investigators and 3,500 staff, and we work together. Our goal is really to change the future of medicine. Our focuses are really 4 things: accelerating research to clinical impact, investing in leading edge technologies. Our particular areas of distinction at this point are cell and gene therapy and big data and omics. Strengthening partnerships, that's with academic organizations and also with industry in order to bring ideas to patients. And finally, securing our core as a leading academic medical center dedicated to training the next generation of scientists who will be able to solve the most challenging problems in children's health.
Thank you, Sue. Mary?
Yeah, thank you. It's also my great pleasure to be here with my colleagues. So chair of the Department of Pediatrics and physician-in-chief of the Children's Hospital. But again, most relevant to today's conversation, I think, is my role as director of the Maternal and Child Health Research Institute. So this is one of 6 institutes in the biomedical field on this campus. We have cancer, cardiac, stem cell, infection immunology, neurosciences. But I think uniquely as maternal and child health, what we do is try to push the whole maternal and child health research agenda across this entire campus so that people in the School of Engineering or, in our new climate, our new School of Sustainability, where they're thinking about climate, really all of the sciences across this campus, we want to get the most brilliant scientists across the campus thinking about the challenges that are facing mothers and infants and children. And a lot of it's infrastructure core. We have a clinical research support office to enable research. We support a number of cores. We have a lot of educational programs, but really the broader mission is to really leverage the entirety of the campus.
Thank you. So in the past, the bench-to-bedside relationship was maybe hindered a bit because researchers and clinicians were operating a bit in their own silos. Huda, I wonder if you could reflect on this a bit, having started on the clinical side. Were there traditional obstacles that you saw that affected this relationship that you tried to overcome as you structured the work at the Duncan NRI?
I think one of the most important things, if we are serious about having physicians participate in research, is to provide physician scientists with the needed protected time for them to do that research, for them to really tool up with courses, with training and new skills so that they become bilingual in the language of medicine and science at the same time. I was fortunate, as you've heard, Ralph Feigen told me, you don't have to come to the hospital for 3 years. Go do the training you want to do. And I think that's not often happening. And the pressures on hospitals, the pressures of people doing clinical service sometimes end up pulling back and forth these physicians into the clinic. So I think the first obstacle we have to overcome is really go back to training physicians as scientists and give them the needed runway, 3-4 years as they need to become really experts, and then they can contribute in a meaningful way. And that's really an important circle. They can contribute to disease-oriented research and go back and make a difference in the hospital.
So I think CHOP has tried to develop some programs specifically that tie clinicians and researchers together. Can you talk about a couple of those?
Yeah, I want to echo what Huda said about the importance of training and methodologic rigor that people get in training, and really breaking down the walls between research and clinical care. So we operate under we hope to bedside to bench, back to bedside circle. And a couple of programs I think that we're investing in that illustrate that. For example, we have a program called Baby Eagle, which is based in our neonatal intensive care units, where Mary and Huda can probably both relate to this. Sometimes there are infants who have multiple problems that are very difficult to diagnose, and they suggest a potential genetic etiology. And so we're employing a program, this Baby Eagle, that will leverage for those kids who have a suspected underlying disorder. It'll be a low-cost, artificial intelligence-driven genomic test with a rapid clinical turnaround time that will target several hundred of the most seen genetic challenges in neonates. And the goal will be to shorten that diagnostic odyssey that these children and their families face. Sometimes it can take, in other studies where people have looked at this, it can take multiple years to get a diagnosis that often then delays intervention, delays appropriate screening. So that's something, again, that bedside to bench, back to bedside philosophy. The other thing that we're doing is and this predated me, we invest in what we're calling frontier programs that bring together really top-notch clinical care, cutting-edge clinical care, and cutting-edge research. And an example of one of our frontier programs that I think is a great example is a Center for Precision Medicine for high-risk pediatric cancers. And this is led by an individual, Ted Leitch. And the story that inspired him was much like Huda's story with the young girl with Rett syndrome, a little 6-year-old girl that was sent home after having failed multiple oncologic treatments, was sent home on palliative care. And in a precision medicine approach to tailor a treatment to her biology and her tumor's biology, they sent her tumor for sequencing and found a novel genetic change for which oral therapy had been discovered in a different kind of cancer for that particular change. And they used it in this young girl who was at that point on palliative care, and her tumors shrank. And she went from palliative care at age 6 to now, she's in college, she's learned to drive, she's living a regular life, arguing with her parents like any young teenager. But it's just an example of the strength of these kind of precision approaches. And we know that in children oncology, there may be as many as 25 to 50% of kids who have these potentially targetable mutations, and maybe less than 10% are currently treated. And we want to expand the proportion of patients who get this testing, make sure it's accessible and equitable. And then once we define these targetable mutations, develop new treatments for these patients who currently have disorders that are considered undruggable or who have terrible outcomes.
Thank you. Mary, can you talk about your perspective at Stanford, and also maybe you make pretty good use of the dry bench in some of your bedside relations.
No, thank you. I just, if I may just expand a little bit on a couple of points they touched upon, because, I think one thing I would add, we also, I think in the ICU setting, I think you mentioned the NICU specifically, in the ICU setting, this very rapid genome sequencing can really make a difference. So we have this ultra rapid nanopore sequencing where you can get it back in 8 hours, and it can truly talk about the diagnostic odyssey. In the ICU, you don't have weeks and weeks and weeks for the diagnostic odyssey. What I want to add to that part is if you're going to do that, or if you're going to do this precision treatment of cancer, where you may very much be treating a mutation and not the traditional cancer, you need a lot of infrastructure within your hospital. You need custom epic order sets. You need a whole new level of investigational drug pharmacy because you have to be so nimble. I think the kind of work Susan is describing, that's absolutely the future, but we have a lot of work to do to enable that. So I wanted to add that layer about doing this kind of work, because regulatory infrastructure, epic order sets, pharmacy, what you just described, Susan, I mean, I've seen these staggering flow charts of what needs to happen depending what path you go down, depending on what your gene mutation is. So I just wanted to add the infrastructure piece being really, really important. And I think, you mentioned the dry bench. I think what we've really tried to do around silos is really trying to break down the silos between not just our departments, but really across all of the schools on this campus. The dry bench, I mean, the possibilities are sort of limitless. Whether, I think we're going to talk about big data, and, you talked a little bit about computational biology, but we have whole other kinds of big data, how we use artificial intelligence. And I think one of the things we've tried to do across all of these disciplines is make sure we're not losing sight of health equity. But that's the other, if you're going to do these precision things, you want to make sure you're really bringing them with it, making sure you're very mindful about the health equity perspective. That's the slippery slope of some of these precision, treatments, is that you want to make sure they're accessible to everyone. I don't know, dry bench is such a broad topic. I'm not sure, maybe you could help me understand a little bit.
Yeah, so I think when you're as you're talking about the social determinants of health, like, for example, kids at risk for diabetes.
That's a great, yes, that's right. I think this is an example where we've tapped into the expertise across the campus using people in the School of Engineering or, in management science and engineering, bringing artificial intelligence to in our kids with type 1 diabetes. We're using artificial intelligence to look not just at their continuous glucose monitors, but a lot of the social determinants of health to help tailor specific interventions to sort of target the kids that are the most vulnerable, use in terms of how we use telehealth, how we do our clinic workflows, how we process these data, how we get our psychologists involved. This has been something we've done with folks in management science and engineering who've never even done anything in the health space. And I think that's sort of one of the fun things about reaching out to these new disciplines, taking disciplines that have never been applied to health and then bringing them to some of the really vexing challenges that we face.
Huda, please add on to those points as you want to.
I wanted to add really an important point to build on what Mary said about health equity. About 4 to 5 years ago, we've noticed that most of the patients who are coming to us with unsolved diseases, they're advocating for themselves. They know they have connections and they come and we solve the problem. We quickly recognize there is a population very poor. They don't know better. They're fearful about what does genetic testing mean. And those are the people who don't have the same kind of access to health, to this latest technology. It's quite expensive. They don't have the type of insurance that might help defray some of the cost. And recognizing that in the city of Houston, we developed something that's called the Texome Project, TEXOME.org. It is for the Texas exome, led by investigators at Baylor College of Medicine and Texas Children's Hospital, specifically to reach out into the communities that's diverse, that's underserved, that doesn't have access, doesn't have insurance, doesn't have any access to such technology to ensure equity. And that has been very popular program and very helpful, really changing the access of healthcare. And we started that way before the big movement and attention to health equity. It started about 3 to 4 years ago. Part of it was funded by the NIH, but there are way too many patients to really, where NIH funds will suffice. So Texas Children is matching dollar for dollar the NIH funding to double the amount of cases. And it's been great. I cannot tell you how many cases were solved by doing this. And importantly, I think there's an educational component to it. And now the communities are engaged in research and value research, are not scared about what does research do to them. So it's building confidence between communities that have been traditionally underserved and scientists. So that I just wanted to add that because we've recognized that, Mary, a long time ago, and the hospital has been really generous matching the funds to do this.
Thank you. I also, when we I want to get into the training of young scientists later on and talk about equity in that portion of the conversation too. You all have been extremely successful. We've started to talk about collaborations, and I'd like to actually break it into 2 parts. Sue, if you talk about for a moment CHOP's ability to sort of tap into University of Pennsylvania, can you talk about how that assists this drive to accelerate bench-to-bedside discoveries, and the other panelists will pitch in?
Yeah, absolutely. We partner extensively with the University of Pennsylvania. It's our academic home. There's recently we make sure that our core resources are complementary across the institutions. We have examples of strengths from both campuses come together. One of our junior investigators doing fetal gene therapy is partnering with one of the adult leaders of the Penn Cardiovascular Institute on a really large collaborative project across the 2 campuses. Another example beyond Penn, especially speaking to what Mary was talking about, bringing different skill sets to the table to collaborate. We have an example of artificial intelligence research at CHOP that was really a collaboration with Drexel University, which is another university in West Philadelphia that has a very strong computer science program. And speaking to the leveraging of big data, we partnered with them to analyze over 53 million electronic notes from more than 1.5 million patients in the repository to define distinct phenotypes using their medical histories to define distinct phenotypes that were associated with developing future disease and looking at their trajectory. So leveraging the electronic health record, they were able to identify from these 53 million notes about 10,000 phenotypes. And these phenotypes were refined enough using the records to be able to distinguish some different behaviors that could have been all lumped together. For example, they could distinguish autism spectrum disorder from other rare neurodevelopmental disorders. And by linking this big data phenotyping with omic data, for example, we see a future in which you can leverage, and some of this is happening already, this deep phenotyping and the novel genetic causes of disease to identify cohorts, like Huda talked about her early experience, like finding Rats patients. So we might be able to leverage the electronic health record, which would be, getting back to the equity question, more equitable if you show up to care. And again, one could question that, but individuals don't have to seek care to be able to be identified by a phenotype and have the opportunity to participate in some of this novel discovery science to help us find causes and eventually, hopefully, cures.
Yeah, Mary, pick up on any of those points, but I also wanted to pick up just focusing on Stanford. You talked about how you leverage the expertise on campus. Different institutions or hospitals have different levels of resource. They may not have a formal institution yet, but I think you've found at Stanford, just informal gatherings can unexpectedly yield great dividends. And I think there's a great story you have of an endocrinologist and a chemist.
Yeah, no, thank you. It's one of my favorite stories. So my new chief of endocrine, who's a PhD epidemiologist, MD PhD epidemiologist, who does complications of diabetes, was at a wine and cheese for new faculty. And they went around the room and they each introduced themselves. And he said, I study type 1 diabetes. And some chemist said, he makes molecules fast and slow. And so David went over to him afterwards and said, can you make insulin faster? He's like, sure. So now they have R01s together, sort of refining the artificial pancreas. One of the things we do to spark these collaborations is we have $200,000 grants that require you have a co-PI equal in another school on campus. And we're very careful about the intent of this mechanism. You can't just say this chemist is running an assay for me. You have to have a scientist in a discipline where the whole is greater than the sum of the parts. And this collaboration will move both of you into a really new and exciting direction. That is the single most favorite thing we do. They are so unexpected, right? A biophysicist can measure the mechanical forces on a trophoblast when we look at implantation for, people with infertility, or the stuff with I could just go on and on and on. They're so unexpected. They're so exciting. They're so fun. And it's great because these people who may not even have been doing something directly in medicine are so excited to think about new applications and new relevance of their research. Those are really, really fun. So I think people find each other, and that's great. But by putting out these kinds of announcements, we get great stuff happening. The other thing is our other institutes that all have seed grants, we say to them, if one of the seed grants you get that you really like is maternal and child health, we'll fund it. And that is starting to elevate the child health profile in all of the institutes across this campus, which has been, I think, a real way to amplify, our commitment to child health.
I'll just jump in. We do the same with Penn. There's a program called Synergy Grants, where you bring folks across the entire School of Medicine campus and CHOP, and also in a program called Penn Health Tech, which is across the School of Medicine, School of Engineering, and Children's Hospital of Philadelphia, where the same thing, if there's child health applications, we'll be part of, funding those things. That's how magic happens.
That's the math, yeah.
Huda, can you pick up on some of these points and perhaps talk about Duncan NRI's relationship with Baylor College of Medicine?
Sure. So Duncan NRI is a freestanding institute, as I said, dedicated to brain disorders with about 35 faculty members now and about 400 scientists working in the building. It's all basic research towards brain disorders, but about a third are physician scientists. So they go back and forth with the hospital. All of our faculty are faculty from Baylor College of Medicine. The institute is supported and built by Texas Children's, but all our grants are via Baylor, all our faculty are Baylor College of Medicine. Because we're brain-centric, we basically create something called Brain Disorders Dinners, where we invite everybody in the Texas Medical Center who's interested in brain research. They could be at Rice, they could be at Methodist, they could be at Baylor, at UT. They come together in a dinner and we pick a topic and we bring scientists and physicians without slides to really discuss the problem, the challenges of the problem, what basic science is happening there, where are the gaps. And invariably, by the end of the dinner, and it's a seated dinner, and there's, relaxing wine, drinks, fruits, you name it. By the end of that, people are walking hand in hand, designing a new collaboration. Those people may not have met otherwise. So we try, before COVID, we were doing these quarterly. We're now initiating them again. The other thing we do, just as you've heard, we have had for several years now these synergy agreements where an investigator from the institute needs to find a collaborator from any of our neighboring Texas Medical Center institution. It could be Rice, where biomedical and neuroengineering is very strong, or it could be the hospital, it could be Baylor, where a cell biologist is doing something unique that can enhance the science. So we have it in the area of mental health disorders because we think these are a huge challenge. We have it in the area, so these are separate grants, of neurodegeneration, Alzheimer, Parkinson, ALS, and then we have it in the area of epilepsies because there's a lot of room there to really combine innovative network work with molecular work with engineering. So I think you're seeing a similar theme here. Each of us approach it a little bit differently, but the idea is bring people together that normally may not be working together.
Yeah, and with the pediatric patient population, you're dealing with such a much smaller universe. So these collaborations are incredibly important. And I'd like to ask each of you just to expand it a little bit now to national and international collaborations. I know you all have many of them, but if you could reach out, Huda has, referenced earlier, reaching out to a single colleague working on important research that she could collaborate on. But there's also larger initiatives. Sue, could you talk about any examples that you want to?
Yeah, I think that, two come to mind across Penn and CHOP. They're part of the ECHO study. It's an NIH study, environmental child health outcomes. And their ECHO is an NIH initiative across the country. I think they're recruiting 30,000 moms, pregnant people, and their significant others, and then the babies, to look at social determinants of health, biology, and how those things affect outcomes. And at Penn and CHOP, 2,500 moms will be recruited and their partners, and then their infants will be followed at CHOP to look at these long-term outcomes. More broadly, internationally, and I'm sure my colleagues have colleagues involved with this as well, there's terrific opportunities like the NCI Cancer Research UK Grand Challenge program, where this is a competitive around the world program for 25 million British pounds. And there's some investigators from CHOP who are one of the 12 finalist teams competing for a child-focused grant in this Grand Challenge. And what they're trying to do is develop degraders of oncoproteins for childhood solid cancers. And the dream, partnering with academics and industry across the world, is to develop oral bioavailable therapies for some of these very difficult to treat cancers. So, by bringing together the best minds from around the world and making it as seamless as possible, I think that's how we leapfrog into the future.
Thank you. Mary?
Well, I think Sue is too modest, because my favorite example of collaborative research in rare diseases is CKID. I can't believe I have to talk about CKID, Sue, but this incredible thing that Sue led, to get, hundreds and hundreds of kids with chronic kidney disease. Each individual site only, maybe I'll have a dozen, but Sue has brought together one of the most impressive efforts to really track very, very important outcomes in these kids and hundreds of kids across 50 some sites. The other example I love is CARA, again, very rare diseases. In the late 1990s, when the biologics were first becoming available, 2 rheumatologists here at Stanford were just incredibly frustrated that there weren't ways to do these clinical trials in children. And now CARA has 550 members around the world. They have extensive funding from many different foundations from the NIH, and 20,000 kids with rheumatologic disorders have been in clinical trials because of CARA. So I think when the pediatric subspecialties, and I've always been so impressed by CARA because above and beyond the clinical trials, they do clinical practice guidelines, they do very family patient centered outcomes research. But I think chronic kidney disease and things like lupus or these rheumatoid conditions are rare in the world of pediatric versus adult medicine, but incredibly important diseases. And leaders like Sue or the folks that launched CARA, it's an extraordinary labor of love that have just involved hundreds and hundreds of physicians around the world. So it's a labor of love, right, Sue?
It is. And that's a true collaboration. So
that is a collaboration.
Yeah.
I think you have sites that might only enroll 5 kids. I mean, it's a lot of work, but that's what it takes. And that engages everyone, and it's incredibly powerful.
Thank you. Huda, can you build on this? The value of the collaboration?
To cover a little bit different twist, just to show the diversity of things one could do. We definitely, in the case of unsolvable, using the fruit fly to solve diseases, we do that all over the world. We have 500 collaborations, China, Europe, you name it. People send these unsolvable cases now. Our team would work on them. But I'd like to really share slightly different, more at a global health level. Texas Children's has worked in Africa, Botswana, and other countries to really help with AIDS, and the impact there has been huge, reducing HIV in children. That has been an incredible program. The more recent one, I'll give you an example in the area of neglected diseases, new vaccines, particularly developing an inexpensive vaccine by Peter Hotez and Elena Maria Bottazzi and their team. That's a partnership, a collaboration was a little bit different kind. They gave the whole strategy using recombinant protein to BioE in India. So they collaborate with industry, but it was all patent-free, licensing-free, no cost. The vaccine, I think, cost in India about $3, which allowed giving that to 100 million children in between India and Indonesia. So these are some examples at a global health level, because I think you've heard about the different diseases. We have quite a few of that, especially in the area of genetics. But I think these also global health initiatives really bring affordable treatments and tackle big diseases in different countries. And Texas Children has been really investing in that quite significantly.
Thank you. In about the 8 or 9 minutes left, I'd like to hit 2 things. One is the issue of funding and also training the next generation of physician scientists. So when you're trying to make the case to government funders, institutional funders, venture capital funders, philanthropic funders about the payoffs of investing in long-term research, as a society, we've been very bad at getting funding for the long term. Everybody wants the short term, payoff. And in fact, in healthcare, if you solve a chronic disease or fend off a rare disease, you're saving a lifetime in healthcare supportive care. So can you all talk about how you're feeling? Are you still against a headwind in trying to make people understand why they need to invest in this translational research? Who did you want to start with this one?
I am passionate about this one, and I am frustrated about this one, because unfortunately, as you know, Liz, the NIH R01, it's still the same for the last 30 years, $250,000 with all the expenses, with the fact it doesn't cover as much. That's number one. Number two, to get physicians to commit a career in research, they need the money for the training time. They need the coverage of their salary. Otherwise, they're going to be pulled. who is going to pay their salary if the grant can't cover them, if there is a particular cap that will never really afford a neurosurgeon being in research or an anesthesiologist? How are we going to do that? We're always going to be missing populations. So I think we need to rethink how we're going to support research to really re-engage physicians, scientists in research, and to grow the biomedical research enterprise, to have all the domains you need to have in it. So I feel this is very important. We're lagging behind in this country. We're going to really suffer if we don't do something now to invest in these people. So I'm very passionate about it, and I hope we will do something about it at a government level, at an NIH level, so that we can afford training these people and afford retaining them in science.
Sue?
Yeah, I echo what Huda has to say. And it's incumbent upon us, I think, to communicate to all the stakeholders, policymakers, governmental officials, when we communicate to potential philanthropists and other, industry, to convey some, I think, the passion that you've heard here today about what we can do and what we see about the impact that we can have, and that how the investment now, and particularly, I think for us on this phone, we would all agree, the investment in child health and even that mom to baby continuum, that investments early on can have enormous impact in the long run. I mean, one of my favorite recent things that I like to talk about is some of our investigators who've been able to show that just, the way how our insides affect our outsides and our life, have been able to show how the microbiome that develops in an infant it's important to make a normal and effective immune system. So interventions that early can have enormous lifetime impact. And I think we all need to be able to speak to that and advocate, as Huda said, continue to advocate for the importance of investing appropriately in these initiatives.
Thank you. Mary?
Well, the timing's perfect because the National Academy of Science, Engineering, and Medicine just did a report on the pediatric subspecialty workforce. I mean, the whole physician scientist pipeline we worry about a great deal, but the pediatric one even more so. And so, they just made of their 4 major recommendations. One was specifically about the pediatric physician scientist. Very concrete recommendations. These career development awards should provide greater salary support because they do require a high salary cost share. Liz, they expect you to be 75% protected, but they don't fund 75% of your salary, right? So the NASM said we need to increase the salary support. You actually need to provide money for the research, and you need to provide money for the mentorship team. The other thing the NASM group did is called for collecting much more intentional data about, really understanding the pediatric physician scientist workforce. So we're just now in the stage when this report came out. The policy briefs are being written. There's going to be a big summit held a week from today. So, exactly what Huda and Susan are calling for. I think we now have the backing of the National Academy as sort of an urgent call to action around this. And hopefully the Pediatric Policy Council, the American Academy of Pediatrics, all of us will come together collectively and speak with one voice about how important this is for the health of children in the future.
Thank you. I'd like to close by talking about the next generation of physician scientists. One quality that has come through all of you in this conversation, I'd like you to talk about what are the characteristics, just very briefly, of what a physician scientist should have, and then where we get them and how to go into traditionally underserved communities to tap talent. But professional generosity and a willingness to collaborate seems to be utterly essential in what you do. It's the patient first and sharing that information. Huda, can you talk about how you are developing physician scientists, what you'd like to see in them?
Yes, thank you. So I do believe until we have broader policies and bigger changes, we can start within our own institutions. So we're committed here to train physician scientists, design the career path that's most efficient for them, supporting them with our own fund so that they can be protected, so that they can do the research they need to do to become really competent. And then through that process, mentorship, because mentoring is not just teaching them techniques or teaching them how to do machine learning and so on and so forth, but it's also how to build a career, how to sustain a career. So we're investing quite a bit in programs that will help them with that. And then from there, once they transition to faculty members, you have to continue that mentorship for quite a while. The other thing, you want to empower them to develop their own network. So introducing them to basic science colleagues, because sometimes having a pair, a neurosurgeon can maximally give you 50% of their time in the lab. They have to operate 50% to keep their license. So they, for science to move at a similar speed as it might move in a basic researcher's lab, pairing those individuals with a basic scientist who would work with them and collaborate is extremely valuable. So this is a model we're also supporting to help some of these specialties that are traditionally have not been as engaged in basic research, bring that important domain and bring people into that domain to solve complicated diseases.
And a quick follow-up, I think you're also trying to reach kids earlier in the schools to let them know about these career pipelines. Can you just briefly, a couple sentences on that?
Oh, absolutely. So we have a variety of programs. There's something called the Biotech Academy for grades 6 to 8 that Baylor College of Medicine is investing in to expose these kids early on for research. We have the Michael DeBakey High School for health professionals. That's grade 9 to 12 that we're engaging with that high school to expose children at that early stage to science and research. And this is a public school, but they basically are exposed, and it's quite diverse. All of these schools I picked are highly diverse schools, contain people of underrepresented groups. So we expose them early on. And then we have a variety of programs where we bring anywhere from grade 6 to 12 into our hospitals, our institutes, have educational programs to them so that these children are really catching the bug of science. And it's amazing at this age. We had a tour two weeks ago on a Sunday for students here. The fact they walked into a cold room was so impressive to them, that there is a cold room. They didn't understand that concept. I mean, little things really turn children on. So we have a variety. We have the SMART program that's now nationally recognized, again, brings people mostly from underrepresented groups in science, have them spend the summer. We give them lectures, and these are in undergraduates, expose them again to science and biomedical research of all kinds. There are quite a few.
Thank you. Sue?
Similar programs across the spectrum. Our goal is to have the workforce of the future represent the communities we serve. I'll just give an illustration of one of our programs. One of my favorite things to do last year, I got to go to the graduation of the CHOP Science Academy. It's a partnership with the YMCA, very local Philadelphia YMCAs. The kids got to come to CHOP once a week for like 6 to 8 weeks. They, folks that work here taught some lessons. They got to, for example, do chromatography on a strawberry. And one of the kids, she said her favorite thing of the whole thing was she got to take a swab and swab the environment and put it on a plate and see what grew. And she said, my favorite thing was I got to take the bugs home. And so it was really inspiring. And one of the kids talked about when he started, he wanted to be a football player, but after the course, he thought he wanted to be a psychiatrist. And I think that was because the Center for Violence Prevention did this thing about bullying as one of the episodes. So anyway, it was truly inspirational. And as they were wrapping up, there was a mom who had her 6th grader in the program, and she had all her other little kids there. And as graduation, the kids got a little white coat, and the little girl was crying as her mom tried to wrap her up to go home. She's like, Mom, but I want to be a doctor, because so she wanted the white coat. So in the you can't be what you can't see venue, these kind of programs like Huda mentioned, I'm sure Mary at Stanford has similar programs, but they're so critical to the future.
Mary, can you close us out with your?
Well, we love I find our graduation, which is held right out here for our program, where we bring first generation low income, largely Hispanic high schoolers here for the summer. The graduation is so jealous. There's a bubble machine and a taco truck. And what I love about the curriculum is there's a simultaneous curriculum for the parents on how do you fill out financial aid applications, how do you apply for college, and there's peer mentoring. So the people who've graduated from this program who are now, you know, on campuses around the country, really elite universities, are mentoring the high schoolers that are coming through this program. My favorite story, Sue, is one of the moms told me when she picked up the kid the first day, her daughter said, "Mom, it's a real lab, like an actual lab with like actual white coats and actual mice." And so they're wonderful. And I thought it was just the best thing for us coming out of the pandemic, because that graduation brought all the faculty mentors together in a way that was so joyous. The work with the high school students is just wonderful. So
thank you. Dr. Huda Zogby, Dr. Sue Firth, Dr. Mary Leonard, thank you for a truly engrossing conversation. I'm not sure whether it's more accurate to say hope drives translational research or translational research drives hope, but it's clear that patients everywhere should feel deeply encouraged by the pace of discovery and innovative treatments that you and your colleagues are making possible. Thank you.
Thank you, Liz.